Hey AP Bio. So today we need to talk a little bit about the breakdown of photosynthesis. I have mine here. Hopefully you're looking at yours. So this is my handy little drawing that I have used to kind of keep it all straight in my head. And I wanted to go through it with you guys, but I'm obviously not here today. So I figured this would be the best way to do it. Also, let's just go ahead and acknowledge that my fingernails need repainting, and I'm sorry that that is the reality that you have to live with for the next couple of minutes. So, I want to talk today about the light reactions and the Calvin cycle and how plants exactly are using solar energy to turn carbon dioxide that we breathe into sugar. Okay, so this is the light reactions in excruciating detail. It is the first of the two processes that the plants are using, anything that photosynthesizes is using to create sugar, okay? So our first step here is we're within the thylakoid space down here, within the chloroplast itself, okay? Remember the little tubey, not tube, they look like cookies that are stacked on top of each other. Those are called thylakoids. And those are inside the chloroplast. So first thing that needs to happen is down here in the thylakoid space, within the membrane of this thylakoid, we need to break some water up. Okay, so the first thing we're going to do is we're going to break water into all its little components. Okay, the hydrogen is going to just stay in this thylakoid space to become part of our concentration gradient that we're going to use later to run an ATP synthase complex. This oxygen is going to join up with another oxygen that has been, when another water over here broke, and it's going to create O2, which is going to be released as a waste product, which I think is always really hilarious. That plants, this is a waste product, they don't even care about it. And then this electron here that we have stolen from this hydrogen is going to go over here into our photosystems where it will get energized. It talks about it in the book, and it's always really annoying, but the first photosystem is photosystem 2, the second photosystem is photosystem 1. It was just named in order of which they were discovered, and sometimes science is like that. So this electron is going to enter photosystem 2, where solar energy from the sun is going to excite this electron within the chlorophyll molecule that's in here. It's going to join in and bond in with that big chlorophyll complex and get excited. And what we're going to have on the other side is this really excited electron. Okay, that is now going to be put into this electron transport chain, just exactly the same as in respiration, where this electron transport chain is going to use the energy of this electron to push hydrogens into this thylakoid space, again generating this concentration gradient. So we have our electron here, and he is really low energy coming out of the electron transport chain. So we are going to put him into another photosystem, photosystem 1, where the light energy will energize him and put him into yet another electron transport chain. Now this secondary electron transport chain is a little bit different because it's not generating this concentration gradient with the hydrogens. It is instead using the energy of this electron and the electron itself to generate an electron carrier called NADPH. And this is going to be needed in our Calvin cycle, which we'll get to here in a minute. But this electron chain is not moving any hydrogens. Its only job is to do this. Okay? So, within this thylakoid space, we have this huge concentration gradient of all these hydrogens, okay? So, we are just going to let them flow back out into the stroma, or the space outside of the thylakoid. And as they move out, they're going to spin this little turbine on the ATP synthase complex, which is going to phosphorylate, or add another phosphate group onto ADP, creating ATP, which you can see better if I turn the page like this. Okay, so our input into the light reactions is water, which we need to grab the electron from, and light. Oh no, speaking of, my light just went out. It's like Jesus knew I was talking about light. So our inputs are water and light, and our outputs are NADPH, and ATP. And we also make oxygen, but it is a waste product in this case, okay? So we are going to take this NADPH and this ATP, and we are going to move them on over into the Calvin cycle, okay? Where they are going to be used at various stages to basically shove carbons together. So the Calvin cycle is really interesting. It is basically just a complete reversal of the Krebs cycle, or the citric acid cycle. The whole deal with the citric acid cycle was to take a big complex carbon, molecule, and to snap it all into its little bitty pieces. This one is the opposite. We are going to take small carbon-based molecules and make them into these big complicated things, okay? Because we're trying to build sugar. So another weird thing about the carbon cycle, the Calvin cycle, is that it has to turn a whole bunch of times to actually generate the sugar, okay? So that's what all of these little things are out to the side. You're generating a lot of these molecules. You have 3 of these things and 6 of these things, and it's because you're only generating 1 per turn, 1 little half sugar per turn, okay? So let's get into it. So we're going to take CO2 that the plant absorbs from the air around us. We really want this carbon, okay? That's really important. So we are going to take this CO2 molecule, and using this lovely enzyme called Rubisco, we are going to jam one of these onto this RUBP, or ribulose biphosphate. That's what that RUB is, ribulose biphosphate. And this is just a coenzyme that is used to do that. So we're going to take this guy and we're going to jam a carbon dioxide onto it. It's going to form this really awkward looking intermediate molecule, but it does not stay like this for very long. It almost instantaneously is going to snap into this 3-phosphoglycerate. Okay, and so we just have our little 3-carbon molecule, and we actually have 6 of these guys at this point. So there's a lot of this stuff, like, there's lots of these guys happening simultaneously, and we're just focusing on one. Now this portion of the Calvin cycle is called the carbon fixation portion, where we are taking these atmospheric carbons and fixing them into an organic form. Okay, so at this point, we're going to use some of that energy that we created in the light reactions to mess this guy around a little bit, and basically adding some phosphoruses here, which we got from here. So we're going to stick some phosphorus here, or phosphate, here, to create 1,3-biphosphoglycerate. And then we're going to use some more of the energy, that electron carriers that we generated, to remove those phosphate groups to create this guy here called glyceraldehyde 3-phosphate, or G3P. Now this is pretty much the basic component to build a sugar. Okay, this can very easily be turned into starch, into cellulose, into just a glucose molecule. This is a very basic sugar component, and so that is what you are going to produce from this process. Okay, so when we've made 6 of these here, we're going to retain 5 of them, but we're going to ship 1 of them away to be used by the plant. This is our product, this 1 per turn G3P, this 1-glyceraldehyde 3-phosphate per turn. So it's not super efficient, and you end up with a lot of like clunky stuff staying in this cycle, but this can be used to build any of your carbohydrates. Okay, so this portion of the Calvin cycle is called the reduction stage, because we're messing around with these electrons. Okay, we're messing with some electron carriers. Now, at this point, we have made some sugar, or our base component for sugar. Now we just need to regenerate that RUBP so that we can start the cycle again up here. So it is very easy. You just take some energy from the light reactions and just kind of snap these boys together to regenerate that ribulose biphosphate, or RUBP. And it's weird. You have 5 of them here, and you end up with 3 of them here, so you kind of smash these G3P molecules up and re-stick them back together as RUBP, which is then ready to interact with Rubisco and start the cycle all over again. So our inputs here, we need carbon dioxide from the air, and we need all of these high energy electron carriers and ATP that was generated from the previous cycle from these light reactions over here. And then what we produce is this molecule of G3P, which is our basic building block for a carbohydrate. So, light reactions, we are harnessing the energy of the sun, Calvin cycle, to use that energy to create our sugar. Okay? And so, that is photosynthesis in a nutshell. It is, in my opinion, a lot simpler than respiration, because you don't have to do quite as many piecemeal reactions. Okay, it's harder to build something than it is to break it apart. So when we break stuff apart, we have to do it very carefully. That way we don't cause fires and explosions and stuff within your cells, which would be terrifying. So this is a little bit easier to kind of wrap my head around, at least. I don't know if that's the case for y'all as well. But hopefully these papers help a little bit into kind of getting straight this crazy big chemical process. So I will see you guys on Monday, and I hope you have a good weekend. Bye!